What if I told you that bacteria, the tiniest of organisms, can communicate, cooperate, and even outsmart our most potent drugs? Yes, welcome to the complex world of antibiotic resistance. This is a realm where microscopic life forms, through sheer survival instinct, develop strategies to evade the lethal effects of antibiotics. Intricacy is the name of the game, as these bacteria employ a range of mechanisms, from quorum sensing and cooperative behavior to epigenetic adaptations and phage-mediated resistance. They also leverage metabolic adaptations, form persister cells, and use sophisticated systems like CRISPR-Cas for adaptive immunity. Furthermore, they engage in horizontal gene transfer, manipulate ecological niches, and even apply principles of evolutionary game theory. All these insidious tactics together make antibiotic resistance a complex and challenging problem to tackle. Ready to dive into the microscopic world of bacteria and their cunning strategies for survival? Buckle up. Imagine a world where bacteria chat and cooperate. Sounds like a sci-fi movie. Well, it's actually a reality in the microbial world. This fascinating phenomenon is known as quorum sensing. It's a method bacteria use to gauge their population size. When a certain threshold is reached, they start to act in unison, like a well-rehearsed orchestra. Quorum sensing is key to bacteria's cooperative behavior. They secrete signaling molecules called autoinducers into their surroundings. As the bacterial population grows, so does the concentration of these autoinducers. When the concentration hits a critical level, the bacteria respond collectively, altering their behavior in a coordinated fashion. This cooperation can lead to the simultaneous development of resistance traits within a population. It's like a group of rebels strategizing to outwit a common enemy. By working together, the bacteria can resist antibiotics more effectively, making them harder to eliminate. So bacteria are not just surviving, they are thriving, thanks to their social networking skills. Bacteria don't just rely on their DNA to combat antibiotics, they have other tricks up their sleeves. Let's delve into the world of epigenetic adaptations, a fascinating mechanism that bacteria use to increase their survival rates when faced with threats like antibiotics. Unlike genetic mutations, epigenetic changes don't alter the DNA sequence, but rather influence gene expression. Consider this. It's like having a book where the words don't change, but how you interpret them does. Now, you might be wondering, how does this work in bacteria? Well, some bacteria can modify their DNA with methyl groups, a process known as DNA methylation. This modification can switch genes on or off, leading to variations in bacterial populations that can, over time, contribute to antibiotic resistance. Another intriguing aspect is histone modification, a process that can change how tightly DNA is wrapped around proteins called histones, influencing how genes are read. It's like bacteria have their own secret weapon, hidden in their epigenetic modifications. Bacteria have allies in the most unexpected places. Even viruses can lend a helping hand. Let's talk about bacteriophages, or phages for short. These are viruses that specifically infect bacteria. But what's fascinating is how they can influence antibiotic resistance. It's a bit like a microbial spy thriller. A phage invades a bacterial cell, and while it's there, it can pick up snippets of the bacteria's DNA. If that DNA carries a gene for antibiotic resistance, the phage takes it along when it leaves to invade another bacterial cell. This is a process known as transduction, and it's one way that antibiotic resistance genes can spread between bacteria. It's a complex dynamic, and scientists are still working to unravel all the details. But one thing's clear. Phages add another layer of complexity to the challenge of antibiotic resistance. With friends like these, bacteria are even tougher to beat. Sometimes the best defense is to lie low and wait for the storm to pass. Bacteria seem to understand this strategy quite well. Now, imagine a group of bacteria under attack from antibiotics. While most succumb, some crafty ones decide to play dead. They aren't really dead, though. These are what we call persister cells. Persister cells are a subset of bacteria that enter a dormant state in response to stress, such as the presence of antibiotics. They essentially hit the pause button on their metabolic processes, making them temporarily impervious to antibiotic action. But here's the catch. Once the antibiotic onslaught is over, these persisters wake up from their slumber. They return to their normal state, dividing and growing to form a new population. And this new population, you guessed it, is now more resistant to the antibiotics that were once a threat. Like a game of hide and seek, persister cells hide from antibiotics only to emerge later, stronger and more resistant. Bacteria have a way of remembering past encounters with antibiotics, and they use this memory to their advantage. Sounds like science fiction, right? But it's not. The key to this bacterial memory is a system known as CRISPR-Cas, a type of adaptive immunity in bacteria. When a bacterium survives an antibiotic attack, it can capture a snippet of the invader's DNA and incorporate it into its own genome in a region known as the CRISPR array. This DNA snippet serves as a genetic memory of the encounter. If the bacterium encounters the same antibiotic again, the CRISPR-Cas system can recognize it and mount a rapid and effective defense. This fascinating system is a powerful tool for bacteria, allowing them to adapt quickly to new threats. It also adds another layer of complexity to the challenge of antibiotic resistance. So, bacteria not only survive antibiotic attacks, but they also learn from them. In the bacterial world, sharing is truly caring, especially when it comes to resistance genes. Now, let's delve into the fascinating process of horizontal gene transfer and the dynamics of the mobilome. These two phenomena are like the internet of the bacterial world, allowing bacteria to exchange genetic material and rapidly adapt to changing environments. Horizontal gene transfer is the process by which bacteria can acquire genetic material from other bacteria without the need for reproduction. This can include resistance genes, allowing a non-resistant bacterium to suddenly become resistant to antibiotics. The mobilome, on the other hand, is the collection of all the mobile genetic elements in a bacterium, including plasmids, transposons, and integrins. These elements can move around within the bacterial genome or even be transferred to other bacteria, spreading resistance genes like wildfire. Together, horizontal gene transfer and mobilome dynamics create a highly adaptable bacterial landscape, enabling the rapid spread and evolution of antibiotic resistance. With the power of sharing, bacteria can spread resistance far and wide. Our bodies are like vast landscapes for bacteria, full of niches where they can flourish or perish. Imagine a bustling city where each neighborhood represents a different ecological niche in our microbiome. Antibiotics are like a sudden storm, wiping out some neighborhoods while others adapt and thrive. The storm of antibiotics doesn't just wipe out the harmful bacteria, it changes the entire landscape. When antibiotics enter our system, they create selective pressures that alter these ecological niches. The resistant bacteria, those with the right adaptations to survive the storm, thrive in these altered conditions. They multiply, filling the niches left vacant by bacteria that couldn't withstand the antibiotic assault. This change doesn't just affect our immediate health. It has long-term effects on the balance of our microbial communities. Our microbial landscape is reshaped, sometimes in ways that make us more vulnerable to future infections. Every antibiotic we take can reshape the bacterial landscape within us. In the struggle for survival, bacteria are playing a high-stakes game of strategy. To understand this, we turn to evolutionary game theory, an approach that helps explain the strategic interactions between bacteria and antibiotics. Imagine bacteria as players in a game where the prize is survival and the rules are determined by the environment. Each bacterium makes strategic decisions, such as developing resistance or remaining susceptible to antibiotics. These choices depend on what the bacterium anticipates other bacteria will do. In a population dominated by sensitive bacteria, a resistant bacterium can outcompete others as it survives antibiotic exposure. However, if resistance becomes widespread, the tables can turn. Resistant bacteria often grow slower and are outcompeted by sensitive ones in the absence of antibiotics. This dynamic interaction, shaped by cooperative and competitive behaviors, contributes to an ongoing evolutionary arms race between bacteria and antibiotics. In the evolutionary game of life, bacteria are proving to be formidable players. We've journeyed through the complex world of antibiotic resistance, from bacterial communication to strategic survival tactics. We've seen how bacteria use quorum sensing to coordinate their behavior, potentially leading to the simultaneous development of resistance traits within a population. We've delved into the world of epigenetic adaptations, bacteriophages, and metabolic adaptations that contribute to the survival and evolution of bacteria. We've also explored the role of CRISPR-Cas systems in bacteria's adaptive immunity and the intricacies of horizontal gene transfer and mobilome dynamics. We've investigated the complexities of microbial ecosystems within the human body and other environments, and how antibiotics can alter these ecological niches. We've introduced concepts from evolutionary game theory to explain the strategic interactions between bacteria and antibiotics. And finally, we've looked at the distinction between antibiotic resistance and tolerance, emphasizing the existence of persisters and the intricate relationship between antibiotic use and the modulation of the human microbiome. The battle against antibiotic resistance is intricate, but understanding these complexities is our best weapon. Thanks for joining this journey through the microscopic world of bacteria.